Discovery of bacteria. It had long been suspected that small creatures exist, which are too small to be seen with the naked eye. But their discovery was linked to the invention of microscope. A Dutch scientist, Antoine van Leeuwenhoek, was the first to report microbes such as bacteria and protozoa. He used a simple microscope to describe bacteria and protozoa with accurate drawings and descriptions, and called these small creatures as animalcules. He firstly observed small creatures in the rainwater, then confirmed these in saliva, vinegar, infusions, and other substances. The progress in understanding the nature and importance of these tiny organisms has been slow. The existence of microbes was further confirmed by Louis Pasteur. Pasteur went on making many discoveries in the field of microbiology and medicine. His main achievements are the development of vaccines for the disease anthrax, fowl cholera, and rabies. He also made significant contributions in development of pasteurization process and development of fermentation industries. He proved that microorganisms could cause disease. Robert Koch formulated the germ theory of disease. He isolated typical rod-shaped bacteria with the squarish ends, bacilli, from the blood of sheep that had died of anthrax. Then he discovered bacteria that caused tuberculosis and cholera. He formulated 4 postulates which are main pillars of germ theory of disease. These are used to find out whether the organisms found in disease lesions is the causal agent of the disease or not. A specific organism can always be found in association with a given disease. The organism can be isolated and grown in pure culture in the laboratory. The pure culture will produce the disease when inoculated into susceptible animal. It's possible to recover the organism in the pure culture from experimentally infected animal. Koch and his colleagues invented many techniques concerning inoculation, isolation, media preparation, maintenance of pure cultures, and preparation of specimens for microscopic examination. Occurrence of bacteria. Bacteria are widespread in their occurrence. They are found almost everywhere, in air, land, water, oil droplets, oil deposits, food, decaying organic matter, plants, men, and animals. Their kind and number vary according to locality and environmental conditions. Some bacteria are always present and contribute to the natural flora. Others are present in specific environments such as hot springs, alkaline or acidic soils, highly saline environments, in highly polluted soils and waters. Structure of bacteria. All bacterial cells invariably have a cell membrane, cytoplasm, ribosomes, and chromatin bodies. The majority have a cell wall which gives shape to the bacterial cell. Specific structures like capsules, slime, flagella, villi, fimbri, and granules are not found in all bacteria. Bacteria range in size from about 0.1 to 600 micrometer over a single dimension. Bacteria vary in size as much as in shape. The smallest, for example, some members of the genus Mycoplasma, are about 100 to 200 micrometer in diameter, approximately the size of the largest virus, pox virus. Escherichia coli A bacillus of about average size is 1.1 to 1.5 micrometer wide by 2.0 to 6.0 micrometer long. Some spirochetes occasionally reach to 500 micrometer in length, whereas Staphylococcus and Streptococci are 0.75 to 1.25 micrometer in diameter. Recently, a huge bacterium has been discovered in the intestines of the brown surgeonfish, Cantharus nigrofuscus. Epilopatium physalcinae grows as large as 600 micrometer by 80 micrometer, a little smaller than the printed hyphen. It's now clear that a few bacteria are much larger than the average eukaryotic cell. Shape of bacteria. On the basis of general shape, bacteria are classified into three categories. These shapes are known as cocci, bacilli, and spiral. Although most of the bacterial species have fairly constant characteristic cell shape, yet some cells are pleomorphic. They can exist in variety of shapes. Exceptions to the above shapes are trichome forming, sheath, stack, square, star-shaped, spindle-shaped, lobed, and filamentous bacteria. The cocci are spherical or oval bacteria, having one of the several distinct arrangements based on their planes of division. If division is in one plane, it will produce either a Diplococcus or Streptococcus arrangement. When cocci occurs in pairs, then arrangement is Diplococcus. Whereas when cocci form long chain of cells, then arrangement is called Streptococcus. When the division of cell is in two planes, it will produce a tetrad arrangement. A tetrad is a square of four cocci. Thirdly, when the division is in three planes, it will produce a Sarcina arrangement. Sarcina is a cube of 8 cocci. When the division occurs in random planes, it will produce a Staphylococcus arrangement, in which cocci are arranged in irregular, often grape-like clusters. Diplococcus pneumoniae and Staphylococcus aureus are some examples of cocci. Bacilli are rod-shaped bacteria. Bacilli all divide in one plane, producing a bacillus, streptobacillus, or diplobacillus. Bacillus is a single cell of bacteria. Streptobacillus is a chain of bacilli. When rod-shaped bacteria occur in pairs, their arrangement of cells is known as diplobacilli. Examples of rod-shaped bacteria are E. coli, Bacillus subtilis, and Pseudomonas. The spiral-shaped bacteria are spirally coiled. Spirals come in one of the three forms: a vibrio, a spiralum, or a spirochete. Vibrio is curved or comma-shaped rod. Spiralum is thick, rigid spiral. Spirosheet is thin, flexible spiral. Examples of spiral-shaped bacteria are Vibrio, Microbiome.